Dihybrid crossings. Before we had a monohybrid crossing when we checked for one trait only, but now we will just see what will be the probability when we cross two plants with two different traits. So one will be for us the color of the flower. Either it will be a dominant pink flower with a dominant allele pink, purple, and the other one is going to be the white flower. That's going to be a recessive gene for the pea plant. The other traits or characters character will be the color of the peas. The dominant color will be yellow, the yellow peas, and the recessive will be the green. So, homozygous dominant, capital P, Y, and homozygous recessive as a parent generation will cross, and they will have the F1 generation. The F1 generation, all of them will have purple flowers and yellow seeds. Now, what will happen when we will cross two F1 generation plants to have F2 generations? So the gametes can line up like capital P with uppercase Y, capital P with lowercase y, lowercase p with capital Y, and two lowercase letters. Now, make the Punnett square. In this time, the Punnett square will be a little bit bigger, and all of the a little bit bigger. So for the horizontal line, gamete 1 will contain the possibilities of like that, and the gamete 2 will have the possibilities of like that, how the gametes can get one allele from each trait. Pulling it down, we will fill out the table. This is tedious, and I know, and we will learn a trick later on how to make it a little bit simple, because in the test you will have no time to do that. So while I'm just doing it, I just realized that that I just made a mistake in the third row. It's getting there. If heterozygous, it's still it will contain the dominant color. Oops, and this is the problem, because I totally forgot that one of the capital Y's will be, so pay attention here at that. So it should be yellow, but anyway. So at this time I would know that you watched the video and listened to the video too. So when we crossed to FY generation, heterozygous for both traits, pea plants, we figured out that out of the 16 different choices, different scenarios, 9 will be purple flowers with yellow seeds. They are those ones. 3/16 will contain purple flowers and green seeds. 3/16 will contain white flower and yellow seed. And here is the part. And 1/6, this is the part. And 1/16 will have only white flower and green seeds. So the overall ratio is 9 to 3 to 3 to 1. This is the overall ratio when dihybrid crossing, when the heterozygous offsprings will cross. Now, the question is, at F2 generation, the question that Mendel had to answer, non-parental combination of the traits appeared. Why is that? Now, this is because of the law of independent assortment. So, that means that one character, that alleles segregate independently of other pairs of alleles during gamete formation. So, when we how to make it shorter? We check for only the color of the flowers independently and the seed color. We do the Punnett square, the easy one, and now we will figure out how many will have homozygous recessive, 1/4 for the P and 1/4 for the Y. So times together, rule of multiplication, because the probabilities are separate from each other, independent from each other. So when we time it, that's going to be 1/16. So another question, what is the chance number of capital P heterozygous for the color of the flowers and homozygous for the seed color? 2/4 times 1/4 equals 1/8. So you apply the rule of multiplication. Another topic that goes with in this section 2, how can we find the genotype of an organism? Sometimes, actually for the recessive homozygous, we know the genotype. But how can we distinguish between homozygous dominant or heterozygous organisms? Like black labs, they can be black in color if they have 2 dominant genes or they are heterozygous. But the brown lab is always homozygous recessive. When we cross these 2 organisms with the unknown genotype for the black lab, if we this is called the test crossing method, just crossing a unknown genotype with a known genotype. If I have a black lab with homozygous dominant and I will cross with homozygous recessive, all of the lab offsprings will be black. But if I have a heterozygous black lab, in this case, half of the organisms will be black and half of them will be brown. The use of the test crossing method is verifying the true breeding organism. The Mendelian heredity deals with the rules of probability, using the probability rules. That's really important. Now, a little bit of general information too. Now, there are some traits that they are controlled by one gene, and one of them is the albinism. The other one is cystic fibrosis, one of the most lethal disease in the United States, I can say that. And the phenylketonuria, it's very preventable disease if we know that somebody has that. Sickle cell disease, and the 2 last one, the achondrodysplasia, that somebody has shorter limbs but normal body ratios, and the Huntington disease. The first 4 is a recessive disorder. That means 2 recessive genes has to be present to be lethal. If the person has 1 recessive gene, we call that person a carrier person. The phenotype is normal, but with 2 recessive genes, the person will have a positive phenotype. That means the person will get sick. The dominant disorders, one gene is present, that means the phenotype is positive, that person will show that trait of in its phenotype. The dominant trait, although don't think that's a better trait. Dominant trait, think about that is it will be seen in the phenotype. So we distinguish in nature wild type traits. The wild type traits are the most common traits that they can happen in nature. Either it can be recessive or dominant, it doesn't matter. And for example, the freckles, they are dominant traits, but they are less common in the population. See, so it doesn't mean that the dominant is a better or most common. And the mutant trait, let's call it, is the less common in nature, that trait. So this is